Today's chapter is on adaptive immunity. So we have three different lines of immune defenses. We have what's called an innate barrier defense and cellular and molecular defenses, and those are our adaptive responses. And then in the next chapter we'll talk about our other adaptive types of defenses like cellular, humoral, etc. So with our adaptive, it comes from innate types of responses. They take longer to mount. Usually takes a few days, more than a week between detection and response. So our immune system will respond then to a particular antigen. So we have memory. So the second time you get exposed to the same antigen, you will have a very effective immune response that will take place. Now we have two types of responses. We have our cellular, and that's T cell mediated, and then humoral responses are those that are antibody mediated. The goal of both is to eliminate any identified antigen, remember it for the next time, so that things will happen a lot faster. Now with our T cells, they will undergo maturation in the thymus, and our B cells are produced and mature in the bone marrow. So an antigen then is any substance that will trigger an immune response. It will also bind to any antibodies that are produced in response to the antigen. Now a lot of times what happens is that cancer cells will frequently make proteins or polysaccharide antigens. So initially our immune system actually has natural killer cells that will take care of any cancer cells that develop. Now a good term to know is epitope, and that's parts of an antigen that are recognized by B and T cells. So it's like right at the end of the antigen here, and that will bind eventually to an antibody. Now when you activate B cells, it's very important for you guys to be aware that they will differentiate into plasma cells. Good thing to know for your test. And plasma cells will then make antibodies. Now T cells can also be activated, and they also have different types of effector cells. So the two types of T cells that you should know are the cytotoxic T cells, and those will actually directly destroy infected cells and cancer cells, and our helpers. And those don't really seek out and destroy, but they coordinate our immune response with the T cells. So please know these three types of helper T cells. T type 1. These guys will activate your cytotoxic cells, macrophages, and natural killer cells to destroy intracellular pathogens. Type 2, stimulate the B cells to make antibodies. And the T regulatory, they will actually control other types of white blood cells, dendritic cells, mast cells, B and T cells as well. So a lot of times they actually help to regulate the beginning and the end of an immune response. Now cytotoxic T cells, when you think of cyto being cell, toxic is something that would kill a cell, these will directly destroy cells that are virus infected, damaged, or cancerous. Now please know that the cytotoxic T cells will release two types of proteins. One is perforin. These will form pores in target cells. The other is an enzyme called granzymes. And what these do is they break down the host cell proteins. They can enter through a pore, and then they break down the proteins. They induce what's called apoptosis, and that is programmed cell death. Now, please take a screenshot of this slide and upload it to the Dropbox. For your exam, you have to know these 5 types of antibodies. For IgG, These are the most abundant. I'd like you to know that. The most abundant. These will also cross the placenta. They will also cross the placenta. IgA. These can be a monomer or a dimer. And these are found in secretions. So secretions, things like breast milk, for instance, your perspiration, etc. IgM. This is the largest. This is the first one made when you get infected. Okay, it's the largest and the first one made. IgE, please know it fights parasites, and it also mediates your allergic responses. Finally, the last one, IgD, that's the one that is bound to B cells. We don't really know a lot about its function. Now, we have 4 classifications of adaptive immunity. We have naturally and artificially acquired active immunities and naturally and artificially acquired passive. When I say active immunity, please think your body has to make antibodies. Again, active, you have to make the antibodies. Passive means that you are given the antibodies. Okay? So if we take a look at some examples then of naturally acquired active and passive, you could actually get some type of an injection with an antibody or with your naturally acquired. If you had it before, then you've already got the antibodies there. Okay? Now in your passive type of immunities, guys, something like passing from mother to baby. Artificially active, you get an injection with an antigen, but if you get an injection with an antibody, then that would be considered a passive type of immunity. So again, naturally acquired active would be if you happen to contract an infection and that triggers your immune response. So you get an infection, that's a natural occurrence, guys, but what happens then is that you have to make antibodies against that. So it's natural because microbes are naturally occurring, active because you make the antibodies. With the artificially acquired active, you are getting a shot or a vaccine or something like that. So it's artificial and it's stimulating your system to make the antibodies. Naturally acquired passive, again, the baby receives antibodies from the mom. So that's the best description of that. And an artificially acquired passive immunity is if you get a shot that contains antibodies. So a lot of times you might have heard of, let's say you get a tetanus shot. A lot of times it's a immunoglobulin shot. You actually get the antibodies in that injection.